Good morning and good afternoon and good evening to everyone. My name is Eric Liebler. I'm Executive Director of the International Headache Society, joined by our expert faculty, Professor Christina Tassarelli and Professor Masoud Ashina. I'll introduce them a little bit more in a second. We're very excited today to present this webinar, and after an extremely brief introduction and a little bit about us, the IHS, we'll get going with today's program, which is to focus on the highlights of the recently completed, very successful International Headache Congress in Seoul, Korea. And then we'll have some brief survey questions at the end and cordially invite you all. And we have people today on the line everywhere from Boston to Bangladesh, from Mexico City to Miami. So it represents what the International Headache Society is, as we want to be a global force for continued education, research, science, development within the world of headache. Our kind of cornerstones are excellence in science, research, based on the expertise of our leadership, as represented today by Professor Tassarelli and Professor Ashina. We focus on the science, on education, on training. We recognize the importance of being independent, always acting with integrity and transparency, and we are member-driven. We represent over 2,000 healthcare providers of all sorts from all around the world. You could be a clinician, you could be a nurse, you could be an advocate. There is a room for you and a place for you inside the International Headache Society. So I want to thank, first of all, everyone who's listening. You've given up an hour of your day today to listen in and advance your understanding of what is currently on the forefront of migraine research and education. So we appreciate your time. It wouldn't be possible without our faculty. I've got the great good fortune and honor of working with both of our faculty members on a day-to-day basis, and I've done that for a while. They're fantastic. It is volunteer what they do, and they have to do this twice today. They have to do it right now at 5 p.m. their time and again later tonight, so that we can offer this education to people at the right time where they live. I also want to support and thank our educational partner, Pfizer Emerging Markets, enabled us to put this on so we could get this message, get this information to people who could not come to Seoul. The program is being recorded and will be available at the IHS website. So, as I mentioned, we have not just distinguished researchers, educators, and clinical investigators, but leaders of the field. Professor Christina Tassarelli is at the Mondino Institute, the University of Pavia, Italy, where she has many leadership positions within the institute itself. She's written somewhere around 350 to 400 peer-reviewed journal articles, chapters, and is the immediate past president of the International Headache Society, where she led us through the meeting in Seoul. So this was her kind of capstone of her 2 years as president. She became president right after the presidency of our other faculty member, Professor Masoud Ashina, who has been at the Danish Headache Center after immigrating there from Azerbaijan. He leads many of their research efforts into the kind of basic science and the mechanisms of migraine that he'll be explaining a little bit about those highlights. And as I said, he was the president of the International Headache Society from 2019 to 2021. And he continues to work with us today as chairman of our Research and Education Committee, or as a Science and Research Committee. So with no further ado, I want to turn it over to Professor Ashina, who will bring you through two of the highlight topics focused in the area of basic science. He will then turn it over to Professor Tassarelli to go through two of the more clinically oriented aspects. Thank you very much. Professor Ashina, over to you.
Thank you, Eric. Thank you for your kind introduction. It's a pleasure, to be here today and virtually, and to give a presentation on IHS Congress highlights. And my job will be focusing on the science part, which I found interesting. There were many interesting abstracts, topics, sessions covering science, cutting-edge science, but it's impossible to cover everything in 20 minutes, so I will just highlight some of the most important ones, in my opinion. So if we move to my disclosures, you can see my disclosures. As I said, I will cover migraine pathophysiology and highlight on migraine pathophysiology. I will start with just a brief description of a migraine attack. According to the International Headache Classification, we have two major subforms of migraine. Migraine with aura, which constitutes about 1/3 of patients, and migraine without aura, 2/3 of the patients. Migraine attacks are characterized by the recurrent attacks of moderate to severe headaches, and the headache can be associated with photophobia, phonophobia, nausea, and sometimes vomiting. In case with aura, patients report aura symptoms, usually visual, focal neurological symptoms, before headache starts. And number of patients also experience so-called prodrome symptoms occurring before aura or before the head pain starts, and also when the head pain stops. So this is just a brief characterization of migraine attacks. Now, when we talk about migraine pathophysiology, we need to mention the trigeminal vascular system, which is an anatomical and physiological substrate of migraine. What we know is that nociceptive transmissions originates from the activation and sensitization of the first-order trigeminal vascular neurons. Their cell bodies are located in trigeminal ganglion. An ascending nociceptive transmission from the trigeminal ganglion is projected to the brainstem and activating and sensitizing second-order trigeminal vascular neurons, including those in the spinal trigeminal nucleus. These in turn activate and sensitize third-order trigeminal vascular neurons in the thalamus, which subsequently relay the nociceptive transmission to the somatosensory cortex and other cortical areas, ultimately resulting in the perception of migraine pain. What we know from the preclinical data, that activation of the trigeminal vascular system leads to the dilation of intracranial arteries, and this is due to release of the signaling molecules such as nitric oxide, pituitary adenylate cyclase activating peptide PACAP, and most importantly also CGRP, the calcitonin-generated peptide. There is an ample preclinical data which shows that activation of the trigeminal vascular system results in antidromic release of the vasoactive signaling molecules, including CGRP. So because of that, many researchers, in fact, focused on the role of the CGRP in migraine pathophysiology, and also the possibility of using the CGRP as a potential diagnostic biomarker. So the first study that I'd like to share with you, presented during the International Headache Society Congress in Seoul, was in fact the presentation done by our colleague Patricia Pozo-Rosic, who presented this as a part of her Cephalalgia Lecture Award. So the study was focusing on the salivary CGRP and whether the salivary CGRP can be used to monitor the different migraine phases. Okay, so that was our objective of this study, and to work and what they did, that they wanted to evaluate that as a temporal profile during the migraine attacks. So this particular study was observational pilot study, and it included young women with episodic migraine and healthy controls. So what the researchers did, they monitor the salivary CGRP like immunoreactivity during the 30 consecutive days and during the migraine attacks. So it's a very, very interesting study. And they considered 6 time points for the analysis. They defined that as an interictal, it's a 72 hours headache-free. Pre-ictal, it is a 24 hours before the attack starts. Ictal, during the headache onset, after 2 hours or after 8 hours. And post-ictal, it is a 24 hours after the attack. And CGRP levels, they were quantified using the ELISA. So in this study, what you see here is the results. 44 women were recruited here, and you can see the differences in interictal salivary levels of the CGRP between patients and controls. Okay, that was statistically significant. And furthermore, they also found an increase in CGRP levels during the migraine attacks, and patients was classified as having CGRP-dependent and non-CGRP-dependent migraine attacks, meaning that there was a group of patients not exhibiting elevated CGRP levels during the attacks, and they constitute about 20% of the patient. So about 80% of the patients, they presented as a CGRP-dependent attacks. So based on this, the authors of this study, they concluded that the salivary CGRP, which interictally are elevated in episodic patients with migraine, usually in the most of the cases increases during the migraine attacks in the majority of the patients. But the authors also recognize that not every attack is CGRP dependent, and maybe this can explain the underlying pathophysiology of migraine, which means that some patients would be CGRP hypersensitive or not hypersensitive. I will show you later some of the studies that we've done, suggesting that it could be a case. And also maybe it can explain also response to treatment, because we know that not every patient respond to anti-CGRP pathway treatments. So this was a very interesting study, which requires also further confirmation from other groups. Now, the next part of my talk is about future targets. So we move forward. Oh, yes. So here what you see, signaling pathways responsible for the genesis of a migraine attack. We are now on the cell's surface, and you see the different receptors and different molecules binding to these receptors. And some of them you can recognize. And one of them is the CGRP. But the CGRP is not alone, and that's why I'm going to focus now on the other molecules. But the question is that how can we study that in humans? Well, in humans, we can provoke migraine attacks using a so-called migraine model. This is quite unique for migraine because it is a self-limiting and treatable condition. So we can provoke attacks and study the migraine in experimental setup. And the attacks provoked are mimics the usual patient's attacks, so they cannot distinguish that. You can do it using the intravenous infusion or by giving the different provoking agents as oral drugs like sildenafil or cilostazol. So you can see that you can observe the onset of migraine, onset of aura and headache, and then through the whole period of experiment, you can do the different studies, such as MRI studies if you're interested in imaging, or you can also collect the blood samples to measure different biomarkers. So using this model, we showed that the CGRP provokes migraine attacks. And this brings again to a very interesting observation, which is relevant to the previous one in which Patricia Rosich group showed that about 80% of patients, they show elevated CGRP levels during the attacks. Here you see that about 70% of the patients are hypersensitive to the CGRP, and about 30% of the patients, they are not hypersensitive. Again, showing this kind of a difference. But the question is that CGRP is not alone, and the calcitonin family is a group of peptides which share structural similarities with the different other molecules, such as calcitonin, CGRP, amylin, and adrenomedin. You can see that how they share the receptors, and receptors are the complex receptors. They are formed by the calcitonin and so-called RAMP, a modifying protein. So this receptor complex is very, very important. And you can see how CGRP can also act on amylin and how adrenomedin can also act on the CGRP. So when we know that the CGRP provokes migraine attacks, the next question will be whether the adrenomedulin can provoke the attacks, thereby suggesting that it could be also a target for future drug. Well, this study showed also that about 55% of migraine patients, they report migraine attacks after intravenous infusion of adrenomedulin. In addition, we also studied the pramlintide, which is an amylin agonist, and it also showed that about 41% of the patients, they report migraine attacks. So they're not as hypersensitive as for the CGRP, but they are sensitive and significantly more compared to placebo, and again, suggesting that CGRP is not alone. There are other in this family of peptides, possible targets for drug. Let's move further to other molecules, and I'm going to focus on the PACAP signaling pathway. Well, in the human body, we have two different isoforms of PACAP, pituitary adenylate cyclase activating peptide, PACAP38 and PACAP27. The only difference is the number of amino acids. I also put here vasoactive intestinal peptide because they belong to the same family, secrecome family of peptides. They share also receptors, as you can see here. They act usually on the VPAC1 and VPAC2 and PAC1 receptor, and the PACAP can also have additional effect on MRGB2 receptor, which is associated with mast cell degranulation. Now, when we give infusion of PACAP38 and PACAP27, we can induce migraine attacks in more than 50% of the patients, suggesting that PACAP itself or its receptors can be relevant for the drug development. If we look at this receptor profile, here we show you again VPAC1, VPAC2, and PAC1 and MRGB2, you can see that there was one drug before tested acting on receptor, PAC1 receptor. That was a monoclonal antibody against PAC1 receptor. And this compound was tested in migraine patients and it failed. It didn't show any efficacy. It was well tolerated, no safety issues, but it was not efficacious. And that was a 2 doses drug compared to placebo. The next question is that if we target the ligand, whether the effect of ligand will be different. So we have 2 anti-ligand PACAP monoclonal antibodies, and they are now under the development from the 2 different companies. And one of them presented this study during the Congress in Seoul, which showed that the 2 doses of anti-PACAP monoclonal antibody was significantly better than placebo, showing in reduction of monthly migraine days over weeks 1 to 4 compared to placebo. You can see that the majority of the patients, as expected, female patients, and at baseline, they have a 16.7 monthly migraine days, and the mean age was about 43 years old. And what is important that this group of patients tested here, they failed with 2 to 4 previous prophylactic treatments, suggesting that this medication has a possibility being used also in the clinical practice in future, if it's confirmed in the phase 3 studies. So the key point of my presentation, interictally elevated salivary CGRP levels in majority of the patients, and they will increase during the attacks, and amylin and adrenomedulin are involved in the genesis of a migraine attack. PACAP is involved in the genesis of a migraine attack, and the phase 2 trial demonstrates proof of concept of monoclonal antibody against PACAP in migraine prevention. With this, I'd like to thank you for your attention, and I would invite my colleague, Professor Tassorelli, to present her talk.
Thank you, Professor Massoud. Very nice presentation. Very on time, so I can use some more minutes, maybe. Okay, no, I'm kidding. Okay, so I will move to my next slides, and here you can see the disclosure of potential conflicts of interest in the past 2 years. And I have 2 topics today. One represents the practice recommendation that for migraine treatment that International Headache Society has been developing over the past year. And you know that there are a lot of guidelines that are already available from different countries. You can see here those that have been published in the last, let's say, 10 years. And the guidelines are very reliable, but the small problem with the guidelines is that they are based on all types of drugs, and they usually include the most recent drugs. While there are some countries where the most innovative drugs, the most recently released on the market drugs are not available, and they can only rely on the essential list of drugs. And this is the essential list of drugs that is published every 2 years by the World Health Organization. And as you can see, it's not you don't have to expect that all the countries, they have at least the essential list of medicine, because you can see that there is a very wide variability from one country to the others in terms of the essential list of drugs that are available. So this could be a big problem. And in Seoul, there was this poster that was presented by Francesca Polledda. And this is about the distribution of triptans, and this is showing the countries that have at least one triptan available. And you can see that there are a lot of countries that are gray. They don't even have one triptan available. And triptan for the acute treatment of migraine, they have been released in the 90s, early 90s. So 30 years later, you can see that half of the world doesn't have a single triptan. So we decided, okay, guidelines are extremely important, but maybe we should, thinking of our global constituency, we should think of a practice recommendation instead of guidelines. Good practice recommendations are overarching principles derived from common sense and established international agreements on ethics and human rights. This is the general definitions. They are not the result of a formal grade process. They do represent expert opinion. So what we decided, we said, okay, we have to target the entire population of the world, and they don't have all the drugs that we have available, for instance, in Italy for the treatment of migraine. So we have to devise 2 levels of practice recommendation, optimal and essential. Basically 2 sets that can be applied in high resource settings or in low resource settings. And this should also be used where it is appropriate by nurses and clinical officers, because you might know that in some countries they don't have neurologists, they don't have expert in headache. And although the distinction between low resource and high resource settings often refers to a high and low income, middle income countries, we know that in high income countries there are areas where we have low resource settings or areas. And so the ambition was to come out with a set of recommendations that were simple to use, that could be uptaken to the wider population as possible. And basically our aim was to improve and harmonize migrant care all over the world. And it was a complex road that we took, but it was not long, because I think that we acted quite quickly. And in Seoul, Francesca Polledda, she already presented part of the output, and this is the steering committee. You can see here myself, Professor Masoud Ashina, Professor Diener, Professor Bernstein, Professor Sacco, Dr. Pozo Rosic, and our executive directors with his 2 dogs, of course. Then we had quite a large panel of senior members, and probably you might recognize here famous people, but we also wanted to involve the junior members, because it is important that juniors are involved. It is important also for their formation. And basically this was the timeline that was presented in Seoul. So already the results presented at the International Headache Congress. The first manuscript draft was completed in August, but show you an update, because in November 2023, we had the final draft of the recommendation that was posted on the website. And we are now finalizing the recommendation and the accompanying paper that will be submitted to Cefalalgia by December 2023. And if it is well accepted and well received by the journal, it could be out, published, and available for the entire world by next January. And this is the paper that we are working. This is the very last version. And I'm giving you an example. So we subdivided the question to which we want to give a recommendation in a question for the acute treatment and question for the preventive treatment. This is just a sample. This, for example, are the question on triptan and non-steroid anti-inflammatory drugs. Then, for instance, this is an example for a preventive treatment questions, a general question on prevention and a focus on the monoclonal antibodies. And this is the structure of the recommendation, how they will be given. So this is the question, for instance, which preventive medication is suggested for people with chronic migraine? And of course, we have two different answers for the optimal level, and we list atogepant, erenumab, betinezumab, fremanezumab, galcanezumab, onabotulinum toxin, or topiramate. On the right side, we don't have all the monoclonal. So we recommend for the essential setting, we recommend the use of topiramate, amitriptyline, beta blockers. All of these are listed on the WHO list of essential drugs. There are other examples here. What are the success criteria that determine whether drug prevention is continued beyond the third month? And we have quite an elaborated recommendation in the case of the optimal level, because we have more option. If we move to the essential level, we state that given the limited number of options available, it is recommended to continue treatment in all patients that report a subjective improvement and who do not have tolerability issue or unwanted side effects. So they are really quite simple. And here again, we thought to bring into the recommendation the peripheral nerve blocks, because in some countries, peripheral nerve blocks belong to the limited options that are available. So do local anesthetic steroid blocks have a place in migraine prevention? We have a type of answer for the optimal setting and a different answer for the essential setting. And we think that this recommendation, they have been a big effort from all of us. We have dedicated a lot of time to them. But I think that they have a very important role, because they exactly fall into the intersectoral global action plan on epilepsy and other neurological disorders, whose vision is to have a brain health valued, promoted, and protected across the life course in all the different areas of the world. And we think that this is very important also because our idea is to use, is to bring the essential level to as many countries as possible. And in those countries, the idea is to use the optimal level to leverage application of and distribution and availability of more drugs where the drugs are not available. This is the first part of my topic. Now I'm moving to the second part of my topics here is the gastrointestinal system and headache. So I'm moving into a turf, which is not my usual turf, but I think it's very interesting. And I try to explain the data that were presented in Seoul to the best of my possibility. And so, for instance, starting from the gut microbiome, let's just focus a little bit on the etymology. And so the words micro and biome, they come from Greek, and micro means small. Biome means life, basically. Microbiota, again, we have a micro, so small, and biota means the living organism of an ecosystem. And when we talk about the microbiome, we have a microbiota that is formed by the microorganisms. They can be bacteria, they can be fungi, they can be protists, algae, and they function they make changes. So they have a theater of activity, and a theater of activity that can be inside the microorganism themselves, where they produce proteins, lipids, polysaccharides, or it can be external. And so they can, for instance, produce signaling molecules. They can deliver toxins. They can deliver organic molecules. So when we say something like, I have a gut feeling, we don't mean simply that we have a sensation in our gut, but we refer to something that is a mental process. I have a gut feeling means that I am feeling something in my brain that could be an emotion, that could be I don't know, a tip, a suggestion, or whatever it is. And we are not making it up, because this is actually what happens. Because you can see that here in this drawing, you can see that the hypothalamus can definitely influence the gut functioning with a different system, efferent nerves, the sympathetic nervous systems, or the release of CRF or cortisol. But it is a two-way road, because you can see that the microbiome in the gut can produce different substances. For instance, the cytokines, for instance, the precursor of serotonin, tryptophan, or neurotransmitters, or even neuropeptides. Professor Messina spoke to you about CGRP. Okay, CGRP can be released by the gut, and it can be released in the circulation, or it can reach directly the brain through the vagus nerve. So definitely we have a system, or two systems here, that are communicating. This is a quite complex slides. I'm just showing to you very simple, because in the next slide, that are the slides that were presented by the speakers on the topic on gastrointestinal system and headache, they are using these words. So we can group the microorganism in different families. The biggest group are represented by the phylum. Then you move to the right, there are the class, order, families, the genus, and the species. So you will see that this phylum, genus, and species will be mentioned in the slides. And so I'm giving you just a view of what has been presented. This is the first speaker. This is Professor Young from the Yonsei University College of Medicine, but you can see he's also the CEO of Microbiotech. And he presented some interesting data. This is not a study that he published, but it is another study, and it's about the gut microbiota in elderly women with migraine. And in this part of the slide, you can see alpha diversity. Alpha diversity represented the microbial community richness and evenness. And you can see that it was evidently decreased in the migrant group at both the genus and the species level. Then if you move on the right side on the slides, you can see also that when you move at the level of the phylum, so it's a big regroupment, there are differences again between the control and the patient with migraine. I'm not dwelling into this, but I just want to show to you that there is a signal, that there is a difference. For instance, this Faecalibacterium is thought to be beneficial, and it is reduced in the migraine group. And here we are at the species level again, so we are at a different level. And here you can see from the significant number here that there are differences between the control and the patient with migraine. And so there is some signal there. We don't know yet what it means, but then Professor Young moved to show the data from his study. And in this study, it was a cross-sectional control study where they evaluated episodic patient with episodic migraine, patient with chronic migraine, and controls, and they evaluated the fecal samples of these patients. And basically what they obtained is that they could identify difference between control and episodic patient with episodic migraine, control and patient with chronic migraine, episodic patient with episodic migraine, and patient with chronic migraine. And you can see here in these figures where you can see that the fold change, the blue or the red one, denotes the difference between the two groups. So in A, control and episodic migraine, B, control and chronic migraine, C, episodic migraine and chronic migraine. They also detected this signal. Basically, they showed that the association between headache frequency and the relative abundance of some types of microbiota, and there was a negative association. So it seems from this finding that having in the microbiota an increased concentration, for instance, of Catenibacterium, the last one, it exposes the patient to have a higher frequency of migraine. So this also could be interesting. So to summarize, there are diversity between the patient with migraine and control. This difference may be of a different way. And this is, of course, it is only initial evidence, but it is initial evidence of altered gut microbiota. And if this were confirmed by additional studies, of course, this will bring in the picture other treatment options, for instance, probiotics, or for instance, fecal transplantation. So this is really interesting. Now I'm moving to another talk that was included in gastrointestinal system and headache. This was delivered by Professor Burali from Turkey. And her talk focused on medication overuse headache. She also showed some very interesting data about medication overuse and food trigger or share the mechanism between the two. I will focus, for sake of time, on medication overuse headache and the possible mechanism. And this is interesting because she showed a model that was used, repeated administration of sumatriptan to the rat. And you can see that studying male or female, there is a reduction in the mechanical threshold when you expose the rats for several days to sumatriptan. Okay, this is not particularly new, but one thing that it is important, and that was shown by this study, is that you treated the animal with minocycline. It is an antibiotic acting on the gut, and it is also an inhibitor of microglia. You can see that basically you inhibit the hyperalgesic situation induced by sumatriptan. So this is again very interesting, and in this study there are also other interesting information that were received, that the treatment with inhibitors of the inflammasome, this BBG injection, is actually inhibiting mechanically allodynia induced by repeated sumatriptan, and also reduces the activation of neurons in the trigeminal nucleus caudalis, and also inhibits the formation of a calcitonin gene-related peptide. This is what is shown on the left, and I thought this was particularly important. Another piece of information that Professor Burali tried to convey to us is that there is a possibility that medication overuse headache, so the repeated intake of acute medication, is inducing a condition of leaky gut. So there are molecules that should stay in the gut that basically they go through the layers of the gut and they can enter the circulation. And this is interesting because she used a model of female spray dolly rats, and she exposed them to chronic 5-week, you can see exposure to piroxicam. And she showed that the chronic use of piroxicam induced a pain-like behavior in these animals. So they were less, they had less mobility. They increased their grooming behavior. They did more head shakes. And this was associated to an increase in the level of calcitonin gene-related peptide in the serum. And What is also interesting is that in this animal exposed to chronic piroxicam drug, there was also an increase of inflammatory markers that are derived from the gut. So definitely there could be a link there. So to summarize, what I've been trying to comment to you is that there are differences in the gut microbiota of migraine patient, and this has been shown. Okay, it has to be confirmed, but there is a signal there, and there is also the possibility that gut-related mechanism, they may explain medication overuse headache. I think this was my last slide. Yes, so I thank you for your attention, and I'm ready to answer questions here with Professor Masoud Ashina.
Terrific. Thank you both, Professor Tesreil and Professor Ashina, showing again your remarkable ability to synthesize a lot of information and present it in exactly the amount of time that was allocated. So we already have our first question, and you guys, there's a comment box, a chat box for all of our guests today. Please feel free to type in your question. Remember, there is no such thing as a bad question. So the first question came in from Ignacio from Spain. So thank you, Ignacio. And it's related to CGRP and the fact that he noted that in one of your slides, Professor Ashina, there was the activity of CGRP being involved in some of the aura phases. And he was curious if you felt that also suggested that CGRP could also be a trigger or be related to CSD.
Thank you very much for a very good question. Yeah, the role of CGRP in cortical spreading depression or cortical spreading depolarization is not fully clarified. We don't have a direct evidence suggesting that the cortical spreading depression as an electrophysiological phenomena can be triggered by the CGRP. But what we have from the human experiments, and this study in fact was also presented during the Congress, IHC Congress in Seoul, showed that if you infuse the CGRP into patients with a known migraine with aura, and about 38% of the patients within the 1 hour will develop migraine with aura. They will develop aura attacks. It is very puzzling. We don't know exactly what is the mechanism, but there is one important fact about this experiment. When we think about aura and in general migraine with aura, we always think about the following sequence. First, no headache, then aura, and then headache. But we know some patients, they may have overlap, while the aura is started and is lasting about 20 minutes, already at 10 minutes, the gradual development of the headache. So the headache can also come during the aura phase. We also know that the patients develop aura without headache, and few patients develop aura attacks in the middle of the headache phase. In case with experiments, when we give the CGRP, it's difficult because all subjects, all patients develop head pain after infusing of the vasodilator, meaning that the first sequence is activation of the trigeminal vascular system and then aura attacks. This is during the experiments. But in real life, and based on the preclinical studies, it is a different sequence. The preclinical studies show that the CST, the cortical spreading depression, can induce nociception, not another way around, meaning that CST is inducing activation of the trigeminal vascular system. But this is a preclinical experiment. In humans, at least here, we see first activation of the trigeminal vascular system and then onset of aura attacks. So it's a very interesting question, but right now I can't answer the exact mechanisms because we need a preclinical studies.
Terrific. Thank you very much. So we've got a second question here. Didn't identify, so it's an anonymous question, but I think one that both of you could provide your thoughts on. So what would you consider the priorities right now in terms of clinical focus, et cetera, for improving care of patients with episodic migraine globally? So this is someone within the Beltway who's kind of focusing or thinking about episodic migraine as a global issue. What would be your priorities?
You want me to start?
Yes.
Sure.
I was thinking that I would have at least three priorities.
All right.
So first thing I would say, educate patients. They need to know what disease they have, because some patient in many countries of the world, the patient having migraine, they don't know they have migraine. So they are not treating as a migraine. So that should be education of the patient, that should know what disease they have. And then the other priority is to bring effective and safe drug to the widest audience possible, because we have drugs, also cheap ones, that are effective. We have the new one, they are less cheap, but we have also the old one that can be effective. So bring the drug to the patient who need them. That would be probably my speaking in a global view.
Thank you. Masood?
So the global view, in my opinion, will be also focusing on migraine, as Christina said, a recognition of migraine, that this kind of recurrent headache disease or disorder can be associated with a significant disability, and to look at the migraine, not just to look at episodic and chronic, just as a migraine. And then remember that all migraine patients should be offered abortive treatment, acute treatment, and the best treatment, in my opinion, would be specific treatment, the targeted therapy, such as the triptans or the gepants or the detans. And in terms of the prophylactics, the preventive medications, I think that episodic patients deserve better, let's say, treatment management, because we're focusing so much on the chronic migraine, but the vast majority of patients, they have episodic migraine, and they're eligible for the prevention, but they don't receive preventive treatments because of they're undertreated, underdiagnosed. In fact, that we're talking about 35% of patients in total migraine patients are eligible for prevention. I'd guess how many of them in US, in North America and Europe, they received, even they're eligible. Not many. So we're talking about between 10 to 20% of the patients. So a lot of patients undertreated, underdiagnosed.
Excellent. Thank you both. So let's travel over to India with a question from Kirat, and I'm gonna mix it in with another question. So Kirat specifically asks, is there any database signal that would help them identify what patient is most likely to respond to an anti-CGRP therapy? And related to that, a little bit on the scientific side, the other question was, are there any practical implications of salivary CGRP to clinical practice? Should we all go out, and if we give one of you a little bit of our saliva, will you be able to tell us if we'll respond to CGRP?
So I guess this question is for me, probably.
For both, but you could start with the salivary part, but then if Professor Tassarelli also, anyone who, if we can figure out obviously how to select the right patient.
Okay, the question is very relevant. The question is about the biomarkers which can predict efficacy of the new therapies. But the question, the next for this will be another question for the audience. Do we need that? When we know that about 60 to 70% of the patients, they do respond, and about 30 and 40% of the patients, they do not respond. And the question is that whether we need anything prior we start the treatment before decide. As I mentioned before, we already have a problem of delivering the treatment to the patients. And if we start putting the other obstacles, such as a measuring of something and then deciding of something will predict the effect, the answer is no. We don't have any good biomarkers for that. And I will discourage of using saliva for this purpose, because this study was a science. It's a paper trying to understand, the pathophysiology of migraine by measuring that. And the question is also whether the production of saliva, which we know that it is both sympathetic nervous system and the parasympathetic, is a good marker for that. So there's many questions about that. So I wouldn't use that. So currently we don't have any, based on the phenotype or other biomarkers, good bulletproof biomarkers, which can be translated easily in the clinical busy practice. So you can use it, to decide whether to give the drug or not to give the drug. We don't have it.
Okay, I agree to disagree with Masoud. Can I?
You can?
Yeah, because I agree with him that we don't have reliable biomarker for now. There are several papers showing that maybe CGRP can be a biomarker of response. Maybe the pain, electrophysiological pain threshold may predict. There are several things, but we don't have. To be honest, we don't have a biomarker. But I think that we would need it. Because having a biomarker of response, knowing beforehand which patient will respond to a drug, I think that will be very important to improve accessibility to the drug. Because Masoud, you're right, 60% of the patients do respond to the migraine-specific preventive treatment. But in several countries, we have to we can use them as a fourth choice drug, not as a first choice drug. So if we had the possibility to say, hey, this patient is going to respond to this drug, why do I have to cycle this patient through beta blockers, topiramate, and whatever it is before providing the possibility to give the patient a migraine-specific medication? So I think we don't have biomarkers. We would need biomarkers. We will get there.
Thanks very much. So I've got one more question here before we begin to move into our closing section very quickly. You guys have been right on time, so we appreciate that. It comes in from Mustafa in Egypt, and it's addressed primarily to you, Professor Tassarelli. Thinking about migraine in children or adolescent migraine, do any of the studies that you're aware of or that you talked about include adolescents or children as well? And in that last part of your presentation about, I've got a gut instinct, do you have any comment or any thoughts on prebiotics, probiotics as possibly having a role in adolescence?
There are some papers published on the use of biotics in children and adolescents, more than adult patients. They are not randomized controlled studies on large populations. So I would not say that the evidence is strong. And with the children and adolescents, we know that we have a big methodological issue because their placebo effect is higher than in adults. So there are some studies with drugs of a proven efficacy in the adult that failed in the children and adolescents because the placebo effect was too high. In one study, at least, was higher than the active drug. So there are a lot of question marks there, but I think it could be a viable approach for children because probiotics are safe. It is believed that the gut of children tends to be more permeable, and so maybe they might be more exposed to having an inflammatory reaction starting from the gut. And so this would predispose to maybe facilitate the inflammatory reaction that we know happens with migraine. So I would definitely consider that a viable option. We need more strong evidence, of course, but I think that some quite strong rationale is there.
Excellent. Well, again, thank you both. One of the final questions that came in, and I'm going to kind of take it on, is what's the single take-home message from IH 2023 that neurologists, practicing neurologists should be aware of? And of course, as you've heard from both of our faculty today, it's that this is an exciting, tremendous time to be involved in migraine, and we need you. Everyone who joined today, migraine is the number one, neurological disease in the world. It affects hundreds of millions of people. And by you joining us, you're getting better educated to help lead the current and next generation of people to understand, as our faculty said, when someone has a migraine, what they can do about it. So in the future, if you'd like to come to some of these events in person, and we know it costs, it's a lot easier to sign up online. But in May of 2024, we're going to have a meeting right in the center of Europe for those of you in or around that area. This isn't our full giant biannual Congress. This is going to be a focused, integrated meeting, about a day and a half, and we welcome you. And then, of course, following our remit of traveling around the world and trying to bring this science to everyone as close to their homes as possible, in fall of 2025, we will be in Sao Paulo, Brazil, for our full International Headache Congress, and we're hoping to, we have very optimistic goals for attendance, so if you're in South America, if you're listening to us, reserve those dates. How can you make it a little bit easier to go? Join the International Headache Society. Many of you are members, but you get access to all of the history of cephalalgia. There are scholarships for juniors moving up in your career, access to another journal, The Neuroscientist, discounts to all of our meetings, and the membership fee, if you're coming from what the World Health Organization calls low or lower middle income, we're going to provide discounts because we don't want anything to stand in the way of your interest in knowledge and understanding to help you help patients and expand the good word about migraine. So with that, there's a couple survey questions that we'd like to kind of get some feel. You can always email us afterwards, but I think my colleagues will put up the first question. This is again, so we can go back and say that we hopefully did a good job and that you absolutely loved the faculty. So I think the survey will come up, Mert. Question number 1. Or I'm supposed to just keep talking until the question comes up. I know that we will be doing this program again. If you want to let some of your colleagues know, it will be being done at, well, it's 5 p.m. in the East Coast of the United States, which is 11 p.m. for both of our faculty members, and other times all around the world. I don't see the survey coming up. And if it doesn't come up soon, there's not going to be any survey because I don't want to keep you here. So let's give it one more second. First question is on. Oh, okay. I guess I can't see it because I don't have that. So first question is up there. How relevant has the meeting been to your clinical practice? We'll give you a couple of seconds to ask it. Obviously, we hope it was relevant, and if not, send us suggestions in the future. So we'll give you about 10, 15 seconds to answer that question. Oh, you must press submit. There's a little green button, and we especially want you to press submit if you've given us a good grade. Let's go to question number 2. And there's only 3, so don't worry. And we won't be reviewing them, so once you answer, you can go on about your day or your evening. So I'm waiting to see the question 2 has come up.
Suspense.
I know it's suspense. I don't even know if the question's there. Because I can't see that. Question 2.
There it is.
There it is. How would you rate this meeting? Okay, now remember, we all have very fragile egos, so we need a good grade. But obviously,
and we cannot see your reply, so feel free to reply whatever you think, because Masood and myself, we are not seeing what you reply. So you're total freedom. Don't be influenced by us.
Total transparency. And again, they reminded me, if you don't press submit, your vote does not count. So this is not like politics these days. You must vote to have your vote counted. And question 3 is now up. Would you be interested in attending a similar meeting or types of meetings like these? We won't repeat the same content, but there is a lot of education to be had out there. And if you're interested, we'll try and find ways to provide more of them. And apparently question 3 is on the screen. You can vote now. Press that green button. Miraculously, we are at like 59 minutes and 5 seconds since we started. And that gives me a warm, fuzzy feeling of having kept on time. So with that, I'm going to thank our speakers, Professor Tassarelli, Professor Ashina. I also do want to thank again our sponsor, Pfizer, for helping share this terrific education. Thank you all for joining, Professor Tassarelli and Professor Ashina. I'll see you later this evening. Thank you, everyone.
Eric.
Great day.
Goodbye, everybody.
Bye-bye.